Welcome back, everyone. I'm AFTD Scientific Director, Dr. Debra Niehoff. The ultimate goal of FTD research is the development of effective treatments. Thanks to recent advances in our understanding of FTD disease mechanisms, new clinical trials are currently underway or are planned for the near future. Our next presenter, Dr. Elizabeth Finger, is an associate professor in the Department of Clinical Neurological Sciences at Western University in London, Ontario, Canada. She's here to talk about the emerging trials in FTD. These trials are bringing growing hope for finding the first disease-modifying treatments for FTD, in addition to better treatments for FTD symptoms. And we're grateful that she can join us here today. If you have any questions during her presentation, please enter them using the Q&A tab you'll see on the screen throughout. After her presentation, Dr. Finger will be joined by Dr. Dianna Wheaton, Director of the FTD Disorders Registry. Together, they will answer as many of your questions about clinical trials and participating in research as they can prior to the end of the session. Dr. Finger, I turn it over to you.
Great, thanks very much, Dr. Niehoff. And I think Dr. Staffaroni's talk nicely set the stage for the building blocks on which we have a lot to talk about now for clinical trials in FTD. First, these are my disclosures. The ones relevant to my talk today are that I do participate as a site in London, Ontario, in several of the FTD clinical trials, some of which will be referred to today, as well as I lead the oxytocin study in FTD called Foxy. So at the beginning, I'll do a relatively brief high-level review of our current treatments and how that sets the stage for emerging clinical trials in FTD, where we'll spend most of our time. At the end, we'll walk through what it looks like to participate in a clinical trial and how you might approach determining whether a trial is right for you. First, we'll talk about two main categories of treatments. The first are symptomatic treatments, and these, just as the name says, target specific symptoms that are problematic in FTD. The second main category are disease-modifying treatments. These do not target a specific symptom, but rather they target the early cellular or molecular processes that end up leading to neurodegeneration in FTD. For our current symptomatic treatments of FTD, most are focused on modulating two neurotransmitters in the brain, either serotonin or dopamine. Typically in FTD, if we see certain behaviors, many of them, such as irritability, impulsivity, agitation, may be correlated with the low levels of serotonin that are frequently seen in the frontal lobes in FTD. The medications that we use were initially often approved for other disorders such as depression, but can be effective because they work to boost the serotonin systems. When behavioral interventions or serotonin-focused treatments aren't sufficient to deal with problematic and especially hyperactive behaviors in FTD, then we often look to medications that reduce or block the dopamine system in the brain. These medications typically come from a class of medications called antipsychotics or neuroleptics. With these kinds of agents and a few others, there is some evidence for effectiveness, specifically in FTD, for certain behaviors, such as agitation, aggression, or compulsive behaviors. However, as many of you will know very well, firsthand, the treatments that we can borrow and use are not always effective for any one patient. And even if they are, often they're modestly effective, and we're still left with a fair bit of symptom burden. Further, for some of the core symptoms of FTD, we still don't have any treatments, not even those that we can borrow that were approved for other uses, to target deficits such as low empathy or apathy that's common in FTD. There are a lot of reasons why there probably are so few proven effective treatments for the symptoms of FTD, and I'll highlight just a couple. And I'd say the field, including myself and many others you heard from today and referred to, are working on these issues. But one problem may be that the symptoms that we use, such as apathy or disinhibition, are pretty complex behavioral descriptions, and they may occur because of different mechanisms of the brain, different neurotransmitters or different networks in the brain going awry. And so it may be that even though we try to treat those as the same symptom, they're in fact different symptoms and need different treatments. Further, we don't integrate much genetic data into our therapeutic approach right now, particularly genes that aren't specific to FTD, but that are related to normal variations in genes affecting neurotransmitter systems, and may mean that one patient responds to a certain medication while another person doesn't. Finally, most clinical trials in FTD to recruit sufficient numbers of patients have to be multi-center, and there's quite a cost in organizing such trials, which can be a rate-limiting step. Despite those challenges, there has been, a little burst, I think, over the past several years of investigators and new ideas for symptom treatments in FTD that's giving us some excitement and hope. Here I've outlined or highlighted just a few that I am aware of. These on this screen are targeting symptoms such as disinhibition and agitation. And at the end of the talk, I'll present a link to the websites where, if you're interested, you can find out more about these specific trials. Some are currently enrolling, and some are ones that we are hopeful or anticipating in the next few years. For symptoms of apathy, loss of sympathy and empathy, we are running the Foxy Trial right now, which is a trial of intranasal oxytocin for these symptoms. The trial's running at 11 sites across US and Canada. We've completed 2/3 of the enrollment, and over the course of the next year, we're anticipating the enrollment of the final 40 patients in that study to determine whether or not that's a useful approach. There are also a lot of creative approaches considering use of smartphones and apps. I've highlighted just one here, and there are others looking at whether apps prompting certain organizational activities or behaviors or chores or social responses might be also useful to help patients with FTD compensate for some of the loss of those skills. For our patients with FTD who have significant language deficits, the field of neuromodulation is taking off as an emerging potential therapy. There have been short, small clinical trials using a couple different types of neuromodulation, TDCS, or the other is TMS, which is transcranial magnetic stimulation. In these therapies, a magnet or an electrical current is placed or run through the surface area of the brain. And depending on the settings, this can cause an excitation or activity of the neurons in that region or an inhibition. And when these approaches are coupled with speech therapy at that time, there have been positive effects over a very short term. So I think where this is headed next is to look at whether these kinds of interventions can produce effects that last days, weeks, or months after the stimulation. We'll now pivot from symptomatic trials and therapies to those that are focusing on disease modification and ultimately prevention in FTD. Most of you will know that at present, similar for Alzheimer's disease, we really have no effective disease-modifying treatments yet for any form of FTD. That is nothing that can delay or prevent the onset, can slow progression, and certainly nothing that can reverse neurodegeneration yet. Some of the excitement in this field has been boosted by genetic discoveries in FTD over the past decade or so that have really illuminated the different mechanisms that lead to neurodegeneration and therefore have highlighted a lot of different potential therapeutic targets. Here are the 3 most common gene mutations that cause FTD worldwide. On the left in orange, we have the MAPTau gene mutation, which causes or is associated with the buildup of the molecule tau in neurons. In blue is the second mutation, the progranulin mutation, which results in the loss of function of one of our 2 copies of the progranulin gene. And this results in about a 50% reduction of the normal levels of the granulin protein in the blood and in the brain. And finally, on the right, the C9orf72 repeat expansion, which is a gene mutation that's the most common cause genetically worldwide of FTD and or ALS. This results in the accumulation of a dipeptide repeat that typically is not present in the cells. There's perhaps the most excitement about the potential to reverse the effects of the granulin mutation. Different scientists and labs and companies are focused on a few approaches, and I'll highlight the general approaches here to hopefully help our understanding of why there's enthusiasm. So one class are the small molecule treatments, and this is perhaps the most familiar to us. These are typically medicines that can be taken in a pill form, and they're made of small molecules that investigators have found can bind something in the cells that then tries to slow down or reverse this neurodegenerative pathway. Another approach are monoclonal antibodies. These are antibodies, so parts of our immune response, that are synthesized, and they're usually given via an infusion to a patient. And the antibodies might be directed towards a certain receptor or a certain molecule to stimulate or block it to affect this cascade. These are typically given about once a month, depending on the treatment. And then finally, on the left, viral vectors are entering now clinical trials, including for the granulin mutation. Gene therapy with viral vectors has been looked at for several decades now in a variety of conditions. And in this kind of approach, a relatively benign virus, often the adenovirus, which is a cause of common colds, is used. The gene that is to be delivered, so a healthy version in this case of a granulin gene or another gene, would be inserted into the virus, and then the virus is delivered to the patient. The virus gets into some of the cells in the patient, and it leaves behind this healthy copy of the gene, the desired gene. And it's then the patient's own enzymes in their cells that read that gene and produce the normal or healthy protein. So this is one way at looking at restoring normal levels of the granulin reduction in patients, and may be useful for other forms as well. For C9orf72, certainly there are trials ongoing with monoclonal antibodies, and another exciting approach, which are antisense oligonucleotides. Antisense oligonucleotides, or ASOs, work at an earlier level. So normally in our cells, we have DNA. DNA, of course, is the code for the genes in our body that gives our cells all the instructions on the proteins that we need for cells to function. Normally, our DNA is transcribed into shorter segments called RNA that are then read by our enzymes, and the RNA then are turned into proteins. Now, if there's a gene mutation, this could be a mutant protein, and with our traditional drug and small molecule approaches, that might be the target of that small molecular drug. In antisense oligonucleotide therapies, a mirror image of the gene mutation in the DNA is created with a sequence of building blocks similar to the DNA. That mirror image recognizes and binds that sequence of mRNA that the gene mutation produced and blocks the translation or blocks the creation of the protein that that gene mutation codes for. So this is thought to be a perhaps very eloquent way to reduce the problem of these abnormal proteins in the first place, to try and block them from even being produced by the cells. This approach has had a lot of success in a neurologic disorder that affects infants called spinal muscular atrophy, and is currently in clinical trials for Huntington's disease as well as ALS. Finally, for the MAPTau mutation, there are serious efforts looking at all of these approaches with different specific therapies, and we're optimistic that some of these will be coming to clinical trials soon. Now, I focused most of my remarks on emerging clinical trials for genetic forms of FTD, but certainly we know, that more than half of FTD appears to be sporadic. The focus on genetic forms is for a few good reasons at this point, as sporadic FTD presents a few additional challenges, several of which Dr. Staffaroni addressed in his talk as well. The main one is that right now, it's difficult, if not impossible, to predict the specific underlying molecular change that a patient with sporadic FTD has. So we know that there are a few common and then many other less common molecular changes that can all present like FTD. And most scientists right now think that effective therapies are probably going to need to be specific to whatever abnormal molecular pattern that patient has. So much of the research, as you heard from Adam, is focused on identifying those kinds of biomarkers that can tell us in patients, whether it's with a blood test, an imaging test, a spinal fluid test, whether their pathology is tau, FTD, TDP43, or some other cause. Once we have those kinds of biomarkers, the second additional challenge is still in making an accurate early diagnosis. So we know, as you will, just as well, and if not better, the early symptoms of FTD are rather nonspecific, and they are seen in much more common disorders such as depression, substance abuse, Alzheimer's disease, or other dementias. So that even at specialized centers, if it's very early in the course of the disease, sometimes it's currently not possible to make an accurate diagnosis. And these kinds of biomarkers will be needed. And ideally, they'll be easy, they'll be economical and accurate, because to identify and diagnose patients early before their quality of life has significantly reduced, we're going to need to be able to test large numbers of patients, most of whom won't have FTD, but may have depression or some other cause, to find those early patients who truly do have FTD and would benefit from these therapies. So for the final part of the talk, I wanted to pivot to discussing what happens in a clinical trial. The first part is to identify, if there is a trial that you'd be interested in considering. And this might be presented to you by your clinician, but we'll point at the end to sources like the Registry and other websites where you can seek out this information as well. After you make contact with those investigators or study teams, you would be invited typically for a screening visit. At the screening visit, you would review with a member of the research team the study consent form. This is a detailed document that has all kinds of information about the nature of the treatment being studied, the rationale for thinking that treatment would be effective, the safety data known to date, the study visit schedule and the procedures that would be performed, as well as information about how your data would be used and the privacy to protect your personal information. Many times, the research team can send you that consent on email or in the mail ahead of time to give you a lot of time to review it in detail, even before that screening visit. But the screening visit represents a really important time to ask all of your questions to make sure that you feel comfortable that it's a good fit for you. If you decide to sign the consent and proceed, often in that same visit, you will then undergo the screening measures. This would typically be interviews and questionnaires about the FTD-related symptoms, both completed by the patient participant as well as a study partner, who's often the closest family member. The visits would often involve blood work, brain imaging tests, electrocardiograms or EKGs to look at heart rate, etc. The results of the screening visit are usually returned to the study site about within 1 month. And at that time, if you meet eligibility criteria, then you'll be invited to go to the randomization and baseline visit. Eligibility criteria will differ from study to study. Some may be looking for a specific severity of FTD because they think that's where the treatment would be most effective. Some may be targeting a specific gene mutation, or some may have exclusion criteria because having certain other medical conditions may represent an increased risk that they'd like to avoid. If you do meet the eligibility criteria and decide to continue, the next step is typically randomization. This is where, depending on the design of the study, you may be randomly assigned to either receive the experimental medication, certain doses of the experimental medication, or a placebo if it's a placebo-controlled study. You then, depending on the study design, would complete interval assessments. These would be similar to what you heard about for the All-FTD study, typically cognitive assessments, behavioral questionnaires, blood work, imaging, heart monitoring, and sometimes lumbar punctures at different intervals depending on the study design, but often every 3 months or 6 months. Most studies so far have been designed to run about 1 year, sometimes 2, so that at the end of that time period, you'd have your last visit in the study. As was asked in the Q&A for the last section, some studies will feature an open label extension, and this really is, it's determined by the sponsor of the study, which is often the biotech or pharma company. For investigator-initiated studies, this is harder because grant funding often doesn't allow an open label, but certainly it's a nice option when it's available. In an open label extension, everyone who participated and stayed eligible for the first part of the study is then invited to continue and get the experimental medication. That's whether or not you had the experimental drug in the first stage or the placebo. It's a chance for everyone who participated to get the study medication. In the ideal setting, the open label extension is continued until the investigators determine that either the drug was not effective, in which case it would be stopped, it was not safe, in which case it would be stopped or would have been stopped earlier if that were determined, or if it does look to be safe and effective, they'll go on to apply for regulatory approval. One important thing to be aware of and think about when considering participating in a clinical trial is what phase the clinical trial is. Their preclinical studies take place in animal models, non-human animal models, to figure out as best we can whether the drug is likely to be safe in humans and whether it's likely to be effective. But of course, studies in humans are essential. Phase 1 trials are the earliest trials in humans of an experimental therapy. It's a small number, usually initially, of healthy adult volunteers and then patients, and the primary focus is on safety. If things look safe in phase 1, the drug may progress to a phase 2 trial with a slightly larger number of participants, additional safety measures, particularly looking at longer dosing of the drug to make sure that's safe, and starting to look at efficacy measures. In phase 3, for medications that looked safe and promising in phase 2, we'll typically see a randomized double-blind controlled placebo-controlled trial, which is the gold standard for determining whether a treatment is effective or not, as the placebo arm is needed so that everyone is objective about whether or not they think it's working. And then finally, as we mentioned, these may go on for regulatory approval and additional monitoring for safety post-approval. Every trial will be a little different, as I've alluded to, and will have slightly different goals, designs, etc. But these are some of the, I think, most important elements to think about and consider when you're deciding, is the clinical trial a good fit for you? As a conclusion, I just wanted to share a little bit of my personal approach. So I am a cognitive neurologist and a researcher, and I see patients in the clinic here in Ontario. And when I'm talking to my patients about clinical trials and their role in their care, my main message kind of boils down to this. You know as well as I do, overall, the long-term course of FTD is not a good one. We don't yet have treatments that can really maintain quality of life in the long term for most patients. So of course, going into clinical trials, we have hope that this will make it better for that patient during that trial and going forward. However, if we're realistic, we know the brain and the science is complex. If we look at Alzheimer's disease, which is a bit further ahead in understanding, they haven't got it right yet. So I do caution my patients that the chance that any one trial is going to be the drug that changes everything is very small. However, we only participate in the trials we're excited about the science for. And so my take home is that the best reason, I think, for patients to decide to participate in a trial is if your main goal, or one of the main goals, is that you want to contribute to finding better treatments for others in the future. Because if that's one of your main motivations, then whether you get randomized to placebo or the drug, whether it works or not, you've achieved that. You've made that important contribution, because we need to get through all of these trials that are really good ideas to find the one that's going to be the answer, or the several that are going to be the answer. So with that, I'll just leave these websites, the NIH for general information about clinical trials, and then clinicaltrials.gov, where you can find some of the studies that I've mentioned, get details and contact information if you want to find out about more, as well as others that I may not have been aware of. At this point, I'd like to reintroduce Dr. Dianna Wheaton, Director of the FTD Registry, to tell you a little bit about how that will further help you understand, navigate, and connect with clinical trials.
Thank you very much, Dr. Finger. I just want to take a brief moment to introduce people to the registry. This is a place where persons diagnosed, biological family members, and spouse and caregivers can actually join either the contact registry to receive emails about FTD and the registry itself. It'll include news and general research updates, or to participate more fully in the research registry, where people will be able to complete surveys to help researchers better understand FTD, as well as receive study notices that are more directly related to their condition specifically. The registry is currently providing study promotion and referral support to 14 external research studies and clinical trials. So the registry is really an important place that can help you connect to research in the FTD space. Thank you.
Thank you, Dr. Finger and Dr. Wheaton, for that information and an excellent presentation. We do have a couple of questions that we will have time to take answers. And the first is, since many of the emerging clinical trials are targeting genetic FTD, should people be tested to see if they have the genetic form or sporadic FTD? And if so, how do they get genetic testing?
Yeah, it's a great question, and there's not one simple answer. I think you would work through this through your clinician or a referral to a cognitive neurology clinic for a more personalized discussion. Certainly, you know, the equation changes a bit. If there are no clinical trials to test, then it may not be worth finding out your genetic status, depending on your specific situation. But if you think that, yep, if I knew I carried the gene, I'd really want to participate in a clinical trial, then that equation about genetic testing changes a bit. So that is really an individual conversation, but I think it's important to be aware that these trials are out there, so that especially if you do have a strong family history where it seems to be hitting every generation, it may be worth pursuing those conversations with your physician or genetics counselor especially. There's an added layer of complexity that's going to come into some of these trials and is present for at least one right now, which is, as Dr. Staffaroni described, it's complex to figure out in someone who carries a genetic mutation when they'll actually start to show symptoms of the disease. And for clinical trials to figure out whether they're effective or not, they ideally really need to focus on enrolling patients who are close to that symptom onset or at the early stages of it. So that for some trials, you may not only need to know that you carry the mutation, but you may need to have other biomarkers, such as levels of something called NFL in your blood or spinal fluid, that show that you are either an early symptomatic stage or you're likely to become symptomatic in the next 2 to 3 years.
And we're almost out of time, but I know a question that's on many people's minds is, do you have any kind of thoughts about when, if the trials were to show that these treatments were effective, when they might be available?
So that process from phase 1 to phase 3 and then approval typically takes many years. Because FTD is considered a rare disorder, there are fast track routes that may be possible through the FDA to accelerate those timelines. But I'd say as of yet, it looks like we're several years away at least, because we haven't yet had anything come out of a phase 3 trial that's shown efficacy yet.
Thank you. And unfortunately, we're out of time. Thanks again to both of our presenters. I hope that everyone listening found that session as informative as I did. At this point, I'm going to hand things back over to AFTD's Sharon Denny for our next session.